Today we are going to do a monohybrid cross experiment. First, take these pure tall seeds and plant them in this pot, and in the other pot plant these pure dwarf seeds. Both are homozygous. Capital TT for tall and small TT for dwarf. These are true breeding or pure lines. Here you can see the result. The seed which contain capital TT gives rise to a tall plant, and the other seed which contains small TT gives rise to a dwarf plant. In the next step, we will use the tall plant as male parent and the short plant as the female parent. But before pollination, we need to perform emasculation on the dwarf plant. Emasculation means removing the stamens. The stamens are the male part of the flower. By removing them, we prevent self-pollination. Here we have removed the stamens. Now the short plant cannot produce pollen. The tall plant here using is a pure breeding and homozygous dominant. This plant we are using as the male parent. So it can produce only one kind of pollen. Each pollen grain from this plant will carry only the dominant T allele. This is our female parent, a pure dwarf plant with the genotype small tt. We have emasculated this plant, so it will function only as a female, producing egg cells. It's a homozygous recessive, so all the eggs it produces will carry only the small t allele. Now gently dust this pollen onto the stigma of the emasculated flower. Now fertilization will occur between capital T and small t. Now you can see all the seeds from this cross are heterozygous, that is capital T and small t. And when we plant them, they will all grow into tall plants because T is dominant over T. Now look at these plants, they all are tall. These plants are F1 generation. Now we will perform self-pollination on these F1 plants. That means the pollen and the egg will come from the same flower. Since the F1 plant is heterozygous, it makes two types of gametes. The pollen produces two types of gametes, capital T and small t. Just like that, the female part or the egg also produces two types of gametes, capital T and small t. So during self-pollination, any of these gametes can combine randomly. That gives us these possible combinations after fertilization. Now let us collect these self-pollinated seeds and plant them. Even though all the seeds came from tall plants, we now see two types of plants in the F2 generation, tall plants and dwarf plants. Let us check the genotype here. The seed which contains capital TT, when you plant it, it grows into a tall plant. In the other seed which contains one dominant allele and one recessive allele, it still grows tall, but it carries the dwarf gene inside. The seeds in which both the alleles are recessive, we call this homozygous recessive. This seed will grow into a dwarf plant. So in this experiment, we got 2 homozygous tall, 4 heterozygous tall and 2 homozygous dwarf. So the phenotypic ratio is 3:1 and the genotypic ratio is 1:2:1.